Hello my dear students. So today in chapter photosynthesis we will study about nature of light. Okay. Now here, as you all know, that quantity as well as the quality of light influences the photosynthesis, and this light energy shows dual nature. So here you can see that during the propagation it behaves as wave form, and on interaction with matter it behaves as a stream of discrete packets of energy, and that is called photon. So the dual nature of light energy, one is formed as wave, another one is a photon. Now what is this quantum? Quantum is the amount of energy in the photon, and here I have drawn the diagram also. You can see, so this one is a visible range. So visible light is only a small part of electromagnetic spectrum. Okay. See. So visible light is only a small part of spectrum. It ranges from 390 to 760 nanometer, from here to here. So this one is a visible range from 390 nanometer to 760 nanometers. Okay. Now here in this case, what happened? The maximum absorption of visible light by photosynthetic pigment occurs in this red area and the blue. So red and the blue region where the maximum absorption of light takes place, and light absorption in the case of the yellow and the orange zone, here is a minimum, very little amount of the light is absorbed by this zone. So this visible light range or the visible light is only a small part of electromagnetic spectrum. Mainly you have to remember that this one is a 390 nanometer to 760 nanometer is the visible range. Okay. Now we will study about the absorption and the action spectrum. Now this absorption and action spectrum. Absorption 1, a curve which shows the amount of light absorbed at each wavelength is called absorption spectrum. This graph shows the absorption spectrum. Here you can see this is the wavelength, and this one is the amount of light absorbed by the chlorophyll A, chlorophyll B and the carotenoid. In this graph you can see that chlorophyll A and chlorophyll B, the maximum absorption is in the region of violet and the blue color and red color, and minimum absorption in the case of the yellow one, isn't it? So this, the curve which shows the amount of light absorbed at each wavelength is called absorption spectrum. The next one is the curve that shows the rate of photosynthesis at different wavelength is called action spectrum. Now in this graph, action spectrum, here this one is the wavelength they have given, here light absorbed, this line shows the rate of photosynthesis, okay, and this one is the absorption. Now here you can see there is a quite lot of photosynthesis activities going on in the region where the absorption is less. Isn't it? This absorption is less, but the photosynthetic activity is going on. How? Because the light energy absorbed by other pigments other than chlorophyll A, okay, means other forms of chlorophyll or yellow pigment or the orange pigment. Whatever the energy absorbed by these pigments, they transfer that energy to chlorophyll A. And that's why the photosynthesis is going on. Fine. So this is the rate of photosynthesis and this is the absorption. So two terms, absorption and the action spectrum. So once again, what is the absorption spectrum? A curve which shows the amount of light absorbed at each wavelength, that is absorption. And the curve that shows the rate of photosynthesis at different wavelength is called action spectrum. Fine. Okay. Now next is experiments. Experiments related to the photosynthesis. And here what happened? Two scientists, that is Van Niel and Robert Hill, that in the reaction of photosynthesis, whatever the oxygen we are getting as the end product, that coming from the water, not from the carbon dioxide. So this Van Niel experiment, the scientist first demonstrated that photosynthesis is essential a light dependent reaction. Fine. So this photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces the carbon dioxide to carbohydrate, because what is the end product of photosynthesis? Carbohydrate, isn't it? And he observed that purple and the green sulfur bacteria, they use hydrogen sulfide and not water in photosynthesis process, and at that time sulfur is set free. So from this he suggested that O2 evolved by the green plants that comes from water, not from CO2. This is the conclusion given by the Niel. Next, Robert Hill. What he did? He suspended the isolated chloroplast. He suspended the isolated chloroplast from the spinach leaves and he put it in water which is without carbon dioxide, that isolated chloroplast. Then he added ferric salt as hydrogen acceptor, and this whole suspension was exposed to sunlight. Then he observed that ferric salt, they are reduced to ferrous and oxygen bubbles are evolving. Then he proved that water splits, that is photolysis of water. Photolysis of water is nothing but the splitting of water molecule. Fine. So whatever the oxygen is evolved, that comes from the water. So these are the two experiments done by Niel and the Robert Hill. Later Dr. Arnon, he discovered that substance present in the chloroplast. Okay, so substance which is present in the chloroplast and that act as hydrogen acceptor, and that is NADP. So this Arnon, he discovered that the substance which is present in chloroplast and that act as a hydrogen acceptor is nothing but the NADP. So these are the few experiments related to photosynthesis they did, and they come to the conclusion that whatever the oxygen we are getting as an end product, that comes from the water. Now next is steps of photosynthesis. What are the steps of whole photosynthesis process is completed in two steps, main and broadly in two steps, that is the light reaction and dark reaction. So one by one we will study about each. Now here the light reaction. Fine. Now light reaction, first of all we have to study that in light reaction, in the earliest video also I have explained that light reaction takes place in the thylakoid, okay, of chloroplast, and dark reaction takes place in the stroma. Now here photosystem 1 and photosystem 2, they are present in the thylakoid. And these photosystem and photosystem 1 and 2, they help in absorption of the light. Photosystem 1 is of 700 nanometer and photosystem 2 is of 680 nanometer. They play very important role in the absorption of light. So here you can see I have drawn one diagram. This shows the ATP synthesis through chemoosmosis. Okay, and as I already said that light reaction takes place in the thylakoid. So here this one is the thylakoid membrane, this dark zone. Okay, this is a thylakoid membrane. This is the thylakoid lumen, inside portion. This is called lumen. You can see this outer one is the stroma. All these parts already I explained in the structure of the chloroplast in my previous video. Now here you can see this one is a photosystem 2 and this one is photosystem 1. Fine. So photosystem 2, here the cytochrome B and cytochrome F is written. Now what happened? As soon as the light is absorbed by this photosystem 2, splitting of water molecules takes place. Here you can see this water molecule that splits into the oxygen and the hydrogen. Fine. So splitting of water molecules takes place. Hydrogen ions accumulate, this one, in the lumen. This hydrogen ions accumulate in the lumen. This is the lumen part of the thylakoid. Fine. An NADP reductase enzyme. This is located in the stroma zone here, where the dark reaction takes place. So for the reduction of NADP to NADPH2, they require the electrons, and that comes from ferridoxin. Okay. So mainly they require the protons for the reduction of NADP to NADPH2. So that reduction required the protons, and which will come from the electrons that come from ferridoxin. Hence, within the chloroplast, proton in the stroma decreases in number. Proton in the stroma will decrease in number, and the proton in the lumen will increase. See, you can see here the proton, they increase in number in the lumen. Why? Because of the photolysis of water takes place when the photosystem 2 absorbing the light. So this create a proton gradient across the thylakoid membrane, because here the proton concentration is high and here the proton concentration is low. So this create the proton gradient, and there is subsequent spontaneous movement of proton will occur. See, you can see here from the cytochrome B6, cytochrome F. From here this proton is moving out from higher concentration to lower concentration, and that will be utilized here for the production of ATP. Here you can see. So that whatever the proton is moving out from the lumen to the stroma is utilized by this one present in the stroma to give the ATP. So final, this is the ATP synthesis through chemoosmosis, isn't it? So ATP production takes place in the stroma zone. And here you can see further that electron is transported to photosystem 1, and that is again comes out NADP, that will be given the NADPH2, that also is forming. So here ATP is forming. So this is all about the chemoosmosis by which ATP synthesis takes place with the help of the two photosystem, photosystem 1 and photosystem 2, and they both are present in thylakoid. Okay, and thylakoid is a part of the chloroplast where exactly the light reaction takes place, isn't it? So this is all about the steps of photosynthesis, in which this one is a light reaction. Okay, now one more term is there, photophosphorylation. Now what is this photophosphorylation? Here this photophosphorylation is nothing when the formation of ATP takes place in the chloroplast in presence of light is called photophosphorylation. There are two types of photophosphorylation. What are those? Cyclic photophosphorylation and non-cyclic photophosphorylation. So here I have drawn this diagram is of cyclic photophosphorylation. So first I will explain this diagram. So as I already said, there are two photosystems. What are those? Photosystem 1 and photosystem 2, isn't it? So in the case of cyclic photophosphorylation, and what is photophosphorylation? Formation of ATP in the chloroplast in presence of light. Fine. So here you can see in the case of cyclic one, this one is a photosystem 1. Okay, photon of light. Now here what happened in the case of this photosystem 1 absorb energy of different wavelengths. Okay, here the absorption of light will takes place by photosystem 1. Because of that, here the reaction will occur, that will initiate the reaction, and specific molecule of chlorophyll A. Photosystem 1 is of how many nanometer? 700 nanometer, isn't it? Photosystem 1. Now here what happened? That will get excited and they expel the energy rich electron. Here you can see. And that energy rich electron will reach up to the ferredoxin through FRS. Now what is this FRS? FRS is ferredoxin reducing substance. So this electron which is released from photosystem 1 is accepted by ferredoxin through FRS, ferredoxin reducing substance. So when the electron reaches up to the ferredoxin, it will further move into the cytochrome B6. Further electron acceptors are there. Cytochrome F, plastocyanin. Fine. And ultimately, after this plastocyanin, from this, the electrons are back to this photosystem. Here you can see this is back to photosystem 700 nanometer. So thus flow of electron is in the closed circuit. Here one thing you can see that between ferredoxin and cytochrome B6 there is a formation of ATP, and cytochrome B6 to cytochrome F there is also a formation of ATP. Two molecules of ATPs are produced by this single cycle, okay, of photophosphorylation. So electron from the photosystem 1 only, that is of 700 nanometer, they absorb the light energy. Electron is excited, that moves toward and accepted by the ferredoxin through the FRS, ferredoxin reducing substance. Later on, the further electron acceptors are there, cytochrome B6, cytochrome F and plastocyanin, and ultimately back to the photosystem. And in between there are the two ATP molecules are also produced. So this is all about the cyclic photophosphorylation. Now here you can see this cyclic photophosphorylation is a significant process as it produces the additional ATP molecule. Now come to the next one, that is a non-cyclic photophosphorylation. This diagram, as you can see, this one is a non-cyclic. Here that is in the Z form, that is also called as a Z scheme. You know that? So this one is a non-cyclic photophosphorylation. Now in this case, you can see here we are using this system. This photophosphorylation include two photosystem. Photosystem 2 and photosystem 1. Photosystem 2 is of 680 nanometer. Photosystem 1 is of 700 nanometer. So here what happened, that this will start from photosystem 2, that is of 680 nanometer. Here the light energy is absorbed through this photosystem, photosystem 2. Fine. And as soon as the light energy is absorbed, the electron is excited. You can see the 4 electrons that are moving out, and that will be accepted by coenzyme quinone, then plastoquinone. Then after that cytochrome B6 and cytochrome F, then plastocyanin. Those 4 electrons are passing, and ultimately 4 electrons they reach up to the photosystem 1, that is of 700 nanometer. Okay, but here you can see little bit amount of, as soon as the light energy is absorbed by this photosystem, water molecule splits, and that gives the oxygen and the proton, hydrogen, 4 hydrogen ion and the oxygen. Fine. And here you can see the 4 electrons are also released. Those 4 electrons will again back to photosystem 2, because you can see here 4 electrons are moving out. So that is again that come back. Now how we got this 4 electron, oxygen and hydrogen? By the splitting of water molecule. And this splitting of water molecule takes place in the presence of manganese, calcium and the chlorine ion. They play a very important role. In the presence of these only the photolysis of water takes place, and by which we are getting oxygen, hydrogen and 4 electron. Okay. Now whatever the 4 electron which are excited from here and moves out, received by the coenzyme quinone, then plastoquinone, then cytochrome B6 and F complex, then cytochrome plastocyanin, and later on received by photosystem 1. Fine. And here you can see in between, that is from cytochrome B6 and cytochrome F, and here ATP molecule is produced, isn't it? One ATP molecule is produced in this case. Now 4 electrons from the photosystem 1 That will, because again this photosystem 1 will absorb light. Here you can see this absorption of light takes place. Then again the electron will excite it, 4 electrons here, that will move towards the ferredoxin through ferredoxin reducing substance, and 4 electron reaches up to the ferredoxin. Here you can see. So when it reaches up to the ferredoxin, what? 4 electron from photosystem 1. Isn't it? Now what happened here? In this case, NADP is present. So whatever the electron is received to the ferredoxin, here, these electrons are taken up by the coenzyme NADP. Okay. Now leave up to here NADP. 4 electrons, they are taken up by the NADP, which are moving from the photosystem 1 to ferredoxin and then to here. Now come back this side. You know that after splitting of water molecule, we are getting oxygen, hydrogen and electron. So these 4 hydrogen will move toward this side and accepted by NADP, and that will give rise to NADPH2. So by the splitting of water molecule, whatever the hydrogen we are getting, that will add up here NADP and it will form NADPH2. Okay, so what we got here? NADPH2. And from where we are getting this 4 hydrogen? That is by photolysis of water. This photolysis in the presence of what? Manganese, manganese, calcium and the chlorine, chloride ion. In the presence of these 3. See, I will show this one. I have written here. Now you can see. Manganese, chlorine and chloride ion. In the presence of this, photolysis takes place. So these hydrogen that will go and add up with NADP and they will form NADPH2. So in this way here you can see in the case of non-cyclic, ATP production is there and NADPH2 production is there. Isn't it? So here ATP will act as energy donor and NADPH2 will act as a reducing power for the reduction of carbon dioxide to carbohydrate during the dark reaction. So this is all about the non-cyclic photophosphorylation. So got it? And here you can see the main difference between the cyclic and the non-cyclic is that in the case of the cyclic photophosphorylation, only one photosystem is involved and there is no photolysis of water. But in the case of non-cyclic one, photolysis of water takes place, and here both the photosystem 2 and photosystem 1, both are participating in carrying out the electrons from photosystem 2 up to the photosystem 1, then up to the ferredoxin, and the formation of NADPH2 and ATP. So here also the ATP formation takes place. In the case of the cyclic also ATP production takes place. So this is all about the photophosphorylation. Fine. Thank you.